Hi, my name is Lily Carpenter, and I'm a junior studying cell and molecular biology in the School of Arts and Sciences. This summer, I worked with two other PURM Award recipients, Audrey and Jake Becker, who are with me today, in Dr. Hansel Stedman's Gene Therapy Lab. Dr. Stedman's lab is developing a treatment for Duchenne muscular dystrophy using AAV and a therapeutic transgene. In March, they decided to add a new project and begin work on developing a unique SARS-CoV-2 vaccine. This new project was the focus of our work this summer. Since we were not able to physically work in the lab, we had to get creative and find a way to get involved remotely. We ended up starting a journal club in which we discussed a new academic paper each week. Journal club included all 9 students connected to the Stedman lab, ranging from high school seniors to incoming college freshmen, sophomores, and juniors. PhD students from the lab also attended, as well as Dr. Stedman and the high school students' mentor, Dr. Lynch. Some of the members of our team only had an understanding up to a 9th grade level of biology, while others have taken higher level courses and others still are professionals in the field. To meet everyone's level of understanding, we went back to the basics and built up from there. This allowed for a complete and well-rounded understanding of the content for everyone, creating a one-room schoolhouse learning environment. Our journal clubs progressed over the course of the summer to include more responsibility for each student. At first, we all read the same papers and came together for questions and discussion. As we grew our foundation from general immunology to more specific SARS-CoV-2 knowledge, we began to work more independently. We all read one paper with each student focusing on a specific conclusion and a single supporting figure. As we became more comfortable talking about the material and analyzing figures, we also became more independent. By the end of our program, we narrowed each week's topic to a specific area of vaccine development, such as the relationship between adjuvant choice and vaccine efficacy, or T-cell function during the SARS-CoV-2 infection. We chose articles of our own finding to present at our club meetings, which resulted in each student leaving with a comprehensive understanding of that topic.
Hi, my name is Audrey and I am a sophomore studying Health and Societies in the School of Arts and Sciences. Because we had such a wide range of background knowledge levels in each journal club meeting, each student was pushed to up their game each week. For older students, we had to understand each topic well enough to be able to explain it to the students that hadn't yet taken a high-level biology class. For many of us, that meant doing extra preparation for each article to relearn the basics of structural biology, T-cell function, and a million other concepts. This pushed us to solidify our knowledge and elevate our expertise. For younger students, each week meant learning these concepts for the first time and using their foundational knowledge to grasp the topic of discussion. On the right hand of the poster, we've included a list of some of our favorite papers we covered this summer. Immersing ourselves in our project and understanding the academic process was certainly more challenging because of the remote nature of our project this year. Despite the challenge, we had many opportunities to learn skills beyond the scope of breaking down complex papers. In the top left, we've included moving graphics of the 3D structure of the SARS-CoV-2 spike protein, as rendered by ICN3D, a software that uses the NCBI database to model proteins and other structures. We also use powerful bioinformatics tools available to the public through NCBI, such as BLAST to align DNA and protein sequences, and VAST to align amino acid residues between proteins in 3 dimensions. A screenshot of our Zoom discussion of the spike amino acid structure is shown on the left. For an even further enhanced understanding of the spike protein structure, we were able to utilize 3D printing technology to print a highly detailed, larger scaled model of the spike protein, which we were reading so much about, pictured in the bottom right corner. We were also able to learn some wet lab techniques without being at the bench by utilizing great databases such as Jove, which has countless video demonstrations of activities we would have been able to perform if we were in person.
Hey, my name is Jake Becker, and I'm a sophomore in the College of Arts and Sciences studying biology. Journal Club was extremely effective for students of all levels to develop their knowledge of biology and to learn to read academic journals. These types of papers are written for experts in the field, and understanding the content, meaning, and significance of each paper is not intuitive. It's a skill, one that has a serious learning curve to develop. Each student excelled this summer through discussing articles with other students of a wide range of education backgrounds. The lab itself benefited too. Since COVID-19 is a novel virus, the consensus in the field is changing rapidly. It is a full-time job just to stay on top of the relevant information. The student team was able to do the work each week to find new papers and present that information in the context of the lab's work. Each week, presentations on a focused We cut 3 hours of work reading into 4 10-minute presentations on a focus topic. Through this, Journal Club expedites the work happening in the lab. An additional benefit of the one-room schoolhouse structure was learning to frequently and rapidly prototype how we should deliver presentations on high-level topics. We read scholarly articles and quickly produced presentations which effectively relayed that information to the lab members who used that knowledge to aid development of our vaccine in the lab, as well as for other students for whom the information was part of their comprehensive learning. Throughout the summer, our journal clubs hosted professionals in the field from a range of universities and corporations. Support from these attendees gave us confidence to continue our work knowing it was valuable and worthwhile. Here in the middle of our poster, we have a testimonial from Dr. Jean Bennett, who is a professor in the Perelman School of Medicine and creator of Spark Therapeutics' Lux Turner, about our journal club. Thanks.
All right, awesome.